Anybody that just sleep. Good night, don't.
Something that it is full.
Please mute your mic so.
I brought my next.
Please, those of you greeting, don't send your greetings to the chat room. Do not send to the chat room. And mute your mic as you join. Mute your mic as you join, and don't say greetings or your name to the chat room. Okay, so good morning, everybody, and welcome to today's class. I'm sure you can all hear me. Good morning, and welcome to today's class. So we started the lecture on Wednesday with a concept of biology, where we defined biology and looked at some characteristics that qualify a thing to be an organism or a living thing, right? And we looked at the unifying themes in biology. And briefly, we may want to recap that. We said biology is the scientific study of life, and the science or biology is the science that studies life. We also said biologists have some set of properties or characteristics that we use to qualify an entity as a living entity or a non-living entity, right? Then we looked at some properties like cells, which is the basic unit of life, and said that all things are made up of cells. And if it has cells, it is a living thing. If it doesn't have cells, it is not a living thing. And we said cells can either be prokaryotic cell or a eukaryotic cell. And we said what distinguishes one from the other is the presence or absence. Please, I am recording the lecture already. Don't ask that I record. Please, I am recording the lecture already. And we said what differentiates a living organism from a non-living, a prokaryotic cell from a eukaryotic cell is the presence or absence of a membrane-bound nucleus and organelles. And we said organisms also are orderly. That is, there is order in organisms, and that they respond to stimuli, and they regulate their environment, and they are able to process and utilize energy. We also said that they reproduce, and we moved on to talk about the levels of organization from the atom to the biosphere, right? That the atom is the smallest and the fundamental unit of matter. And we said it consists of a nucleus and electron, where we say molecule is two or more atoms that are chemically bound together. And I remember asking you to name that chemical bond that exists between atoms. For instance, in carbohydrates, you have carbon, hydrogen, and oxygen as the atoms, and that is a chemical bond between them. In what's it called? In protein, we also have carbohydrates. We have we have carbon, we have hydrogen, we have oxygen, we have nitrogen, and some sulfur as well. And all these atoms are chemically linked. Then from molecule to organelles, we said organelles are microscopic structures, tiny structures that help the cell to carry out certain functions, right? Like transporting, like this garbage disposal, making proteins, making food, producing energy, and all of that. Those are organelles that do certain things for the cell. Then we said the cell is the fundamental unit of life. And we moved on to look at tissues, which are cells combined to make tissues, organs, related tissues that perform certain functions, then to organ system, to organism, to population, which is a group of organisms of the same species that are capable of interbreeding, that are found in a particular place at a given time. Then community. Please, if you greet in this chat room again, I will remove you from the lecture. You can go and join YouTube. Please do not greet when you come in. Who are you greeting? Who did you see? Alabosi hypocrites. All right. So population, group of organisms of the same species that are able of interbreeding. Then we say community comprises several populations. Like in your room, you have is a community that has the population of cockroach, of rats in your room, of spider, and you, the owner of the room, but there are some tenants that do not pay that occupy the room with you. Then we have ecosystem, which comprises the living and non-living components, right? So we qualify the living as a biotic and the non-living as the abiotic. Then we moved on to look at this organization in a flowchart, how they relate or interrelate. Then we moved on to unifying themes in biology. We said there are certain themes, that there are certain concepts that define biology, right? That may be recurring factors, right? Like cell system. Then we look at cell, that is basic and fundamental structural units of life, prokaryote and eukaryote again. Then we said we use this to illustrate cell. Then we stopped here, slide 2. We said all cells, all levels of life have systems of related parts, right? So what we mean by that is that a system, like, we are not looking at it at the system level, now, like different systems that come together to function. Like, for instance, if you have a car, that car has different things that do different parts that do different things in it, right? Like your tire, your gear, your accelerator or throttle, and what have you, right? All of these come together, right? Related parts that come together to function, right? So we said that a single heart cell, that heart cell, some of you are now saying that is at a cell or system. We're looking at the cell, the heart cell here, as a system that has different parts, right? In that heart, you have different parts that do different things, right? That is what we mean by there is a related, there is some of systems that organisms work in. They work in systems. Then the reproductive system, the ecosystem, and what have you. Then we're starting the class from here today. Structure. In biology, we said that structure and functions are related, right? What we mean by that is that the structure fits function. structure fits function. That is, what a particular structure does in an organism is well structured in such a way that is able to do what it is designed to do. So the structure determines the function. The structure is the shape, right, of the object, while the function is the object's specific role. For instance, your pinnae, your auricle, your hair, that part of the hair, the way it is shaped is so that it can collect air waves, right, and transmit it for transmission. So your ear is so strong. Imagine if your own pinnae is facing backward, right? It's not facing the front, like it's turned the other way. So it will not be able to collect air waves and transmit. And so the structure is so designed, right, to in such a way that it fits certain purpose. Structure and functions are related, right, in biology. Functions and the structure and function are related. Then if you look at this organism that we depict in this picture, you see that it has different structures. Look at that covering, like maybe that chitinous exoskeleton, right? The way it is formed is such a way that it helps the organism to do certain things, right? Maybe to protect, right? It's going to be to protect, right? To shield, for protection, and for other function. Then if you look at the leg, your leg is so designed in such a way that it helps it to walk. If you look at what's it called, duck that has webbed digits, it allows them to do what? To swim in the water. If you look at the birds, their wings are designed for flight. So the structure of a particular structure in an organism is so fitted such a way that it is able to do certain functions. So the structure of different cells differ because they perform different tasks in the body. So structure and function in biology are related. The liver is so structured in such a way that it will be able to perform its role in the body. So in biology, you said structures fits function, or structure and function are related. So what we mean by that is that the structure, right, in an organism, a certain structure is so designed in such a way that it will be able to perform its role that it's designed to perform. Then another unifying theme in biology is there are emergent properties, right? As you moved from the hierarchy, from the different levels of organization, there are emergent properties. That is, there are novel properties that you observe as you move from cell to tissue, from tissue to organ, right? There are new properties, novel properties as you move along the hierarchical organization, right? So with each step upward, there are emergent properties. These properties are new. You can novel properties or emergent properties. That is, new properties that are emerging at each level. As you move from cell to tissue, for instance, there are properties that you see at the tissue level or at the organismal level that you will not find at the lower level. So there are emergent properties, right? So life emerges through organization of various levels. With each step upward in the organizational level, novel properties or emerge, or emergent properties as a result. For instance, an organism can achieve more than what an individual cells could achieve because of the new properties that has emerged as you move from a lower level to the higher level. You will agree with me that as you move from the lower level, for instance, as you move from cell to organism, for instance, there is increase in complexity, and there are emergent properties. There are new properties that are emerging as you move from one organizational level to the other. So that is what we call emergent properties in biology as a unifying theme. One of the unifying themes in biology, right? There are properties, there are new properties that arise as you move from one level. For instance, from the tissue level to the organ system level, from the organ system level to the organism level, there are new properties that that comfort. And as you move along, those that are higher are able to perform certain things better than the lower level or cadre. Okay, we also have this figure illustrates the different levels of organization from biosphere down to atoms, and you will agree with me that there are different properties. For instance, for population, right? Population of organisms, we have certain things that they can do that an individual organism cannot do. For instance, if we come together as a team, we'll be able to do certain things than maybe one person is doing. We can achieve more. So those are the property. We achieve certain properties, maybe more thinking faculty, more strength. We can strategize better than maybe one person that is performing the same task with us. So these are emergent properties as a unifying theme in biology. Then reproduction and inheritance is also one of the unifying themes. All organisms reproduce, right? Like we said earlier, as one of the characteristics, organisms reproduce, and they give birth to younger ones of their kind, right? And the hereditary information is encoded in the DNA. So the DNA is a blueprint of life. The DNA contains the hereditary information, right? In forms of nucleotide bases, your adenine, your cytosine, your guanine, your thymine in DNA, right? And this information, the way it is encoded in the DNA, needs to be translated to a form that the cell can read and interpret, like letters. For instance, your letters are your nitrogenous bases. Your T, your H, your E has to come together to form a D. The word is what the cell can read. But if the letters are scattered everywhere, so they need to come together and present themselves in a readable format. So also the information that is encoded in the DNA has to be presented to the cell in a readable form. So that is why the information needs to be passed through DNA to RNA. And the type of RNA we're talking about is mRNA or messenger RNA. By the time we go to biomolecules, we will delve deeper into DNA and the types of mRNA. Then to protein, how DNA is transcribed to RNA and RNA is translated to protein. So there is reproduction and inheritance, and so this shows the DNA structure and the different nucleotide bases, the A's, the C, T, A, T, T. And you said DNA is genetic material that carries biological information from one generation to the next. So the DNA helps us to transfer information, right? If you say something, say it's in the DNA, it's in the gene, right? It's because if you don't have it, you cannot express it. The DNA, for you to exhibit a certain trait, right, you must have it encoded in the DNA. For instance, if you are tall, there is tallness gene in your DNA. If you are short, you have the short gene in your DNA. If you are intelligent, anything, you can use your right hand to write. Your skin color, everything is coded for by DNA, right? And it is hereditary. You inherit it. As you inherit them from your parents, you express them. And you will do more of that also in this course in the aspect of heredity when we get to cellular reproduction and heredity. Okay, another unifying theme is homeostasis. Homeostasis simply means the maintenance of constant internal conditions. Maintenance of a constant internal condition. That is, there's supposed to be a balance and equilibrium in the internal environment. So homeostasis is that process that regulates or brings about the stable internal environment. And how this comes by a negative feedback. What I mean by a negative feedback is that there is the response, for instance, a response is reducing the initial stimulus. What we mean by that is that, let's look at this picture here, right? When during hot temperature, right? The hot temperature is the signal or the stimulus. Blood flows through the screen, the tiny muscles expand, the sweat glands release water to cool the body. So the hot temperature over there is sending a negative feedback that your temperature is too high, you need to cool down, right? So how does it do that is what is illustrated here. There is blood flow to the skin, the tiny muscles expand, then there is release of water from the sweat glands to cool the body. So we call that the negative feedback, a negative feedback loop, whereby the response, right, reduces the initial stimulus. Your stimulus here in this scenario is the body temperature that is high. So when the body temperature is high, there is a negative signal to the system that my temperature is high, please cool it down, right? So that is how organisms maintain homeostasis to be able to survive different types of environments. And if this fails, then problem arises. So that is also one of the unifying themes. Homeostasis, maintenance of a constant internal environment or condition, whereby there is a negative, we said this occurs through a negative feedback in which the response reduces the initial stimulus. The response is reducing the initial stimulus. The stimulus is when there is a high temperature, right? The response is that the temperature needs to be cooled down. So thereby there is maintenance of the. There are other examples that the system gives a negative feedback that helps to maintain stable internal environment. So this is supposed to be number 7. Then we also have interactions as another feature of as a unifying theme in biology. See that organisms do not just exist in silo, right? Just as you don't exist in silo, you cannot survive without we depend on one another. That is interdependence of organisms in this wise. That is, from molecules to ecosystems, interactions are important in biological systems, right? If you don't interact, for instance, if you don't interact with the air, how will you survive, right? If the plant does not interact with the sunlight, how does it manufacture food, right? And if the goat does not interact with the plant, how will the goat feed, right? So these interactions are really important in biology. And the branch of biology that studies that interaction is ecology. So ecology studies interactions and interdependence of organisms or between organisms, maybe organisms of the same species or organisms of different species in an ecosystem or environment. And it could also be, this interaction could be beneficial, it could also be harmful, right? But in biology, there is interrelatedness, interdependence of organisms. Organisms depend on one another for survival, right? If you look at this tree, this is an ecosystem comprising biotic and abiotic factors, living and non-living things. You can see the elephant. The elephant is interacting with the plant by feeding, right? Then when that animal feeds on that plant, it releases what again? Waste. It excretes and provides nutrients for other plants to grow as well. Then the plant itself absorbs light energy from the sun to release oxygen rather, for animal to breathe. The animal in turn releases CO2 for the plants to take in or to breathe in rather. So you can see there are different levels of interactions as you move among organisms. There are different interactions even between organisms, like I said earlier, organisms of different species, organisms of the same species. There are interactions, and it's also a unifying theme in biology. As a recap, today we started the class with by continuing on what we did the last time, looking at interaction at the unifying themes in biology. We mentioned that there is structure fits function in biology. There is interrelatedness between structure and function. That is, what a structure does fits its certain purpose. In the structure fits purpose in biology. That is, what something does in an organism is directly related to its structure or form. We also said there are emergent properties in along the level of the levels of organization. That is, new properties emerge as you move at each level, right? That we said that novel properties emerge as a result of interactions among components at lower level. We said that an organism, for instance, can achieve more than individual cells could achieve. That is, the more we are, the better suited for us. We also said that there is reproduction and inheritance. That is, organisms reproduce and they transfer hereditary material, right? Encoded in energy, and that the heritable information is encoded in the DNA, and it has to be translated to a form that the cell can utilize, and that is DNA to mRNA to protein. We also said homeostasis, whereby the system regulates, maintain constant internal environment by giving a negative feedback to the system. For instance, if you are hot, your brain sends signals that you are hot, please, and other reactions take place to be able to cool down the body temperature. I will mention interdependence of organisms, whereby organisms depend on one another in an environment. That is, one way or the other, we need one another. You cannot survive alone. Even on highland, you still interacts with those things that are there so that you can be able to survive. Okay, the last one is evolution. Evolution, we say evolution is the core theme of biology because there's no way you want to study biology without mentioning the origin of life, right? So evolution deals with the change in living organisms over time, right? That is, evolution tells us how organisms that we have now are different with organisms that once existed. That is there any relation between organisms that we once had and organisms that we have now? How do we differ from what we used to have? How do present day organisms differ from what we used to have? Do they have any similarity? Do we share anything in common? And the differences that exist between us, that is what evolution deals with. And how are we able to adapt? Natural selection confer adaptation on organisms. So evolution can occur through natural selection of adaptations. Adaptations are beneficial inherited traits that are passed down to future generations. So evolution by natural selection simply means that nature selecting those properties, those traits that help an organism to survive. And once they acquire those traits are heritable, those traits are passed down to their generation. If you look at this, the figure that we have here, you will see that the figure on the left-hand side, that one is prone to attack, right? But here, the one on the right-hand side has developed certain features that you may even find it difficult to tell. Do we have an organism here when you compare with this, right? So what happens there is that there is adaptation for survival, right? And that adaptation is passed down to future generations by this organism. And that is what evolution does. We now have new species over time. We have new species over time as a result of evolution. So the evolution also accounts for the unity and the diversity of life. In unity, maybe because we share common ancestor, right? So there is similarity with that. For instance, humans have more similarity, about 95 to 98%, than what chimpanzee and gorilla. So begin to say, okay, what can account for that? Or what could account for that? Is it because we share common ancestor? Right? So all of these are what evolution teaches us, and we are able to learn from evolution. Then the diversity of life, the diversity of organisms. Okay, even organisms of the same species, how do they differ? For instance, if you pick two living things, two human beings, for instance, you will see that in some aspects, no two humans are the same, right? No two humans are 100% identical. No two humans are 100% the same. So that is the diversity in and also unity. So evolution accounts for the diversity and the unity of life. That is, in terms of unity, do we have things in common? And I said that yes, every organisms have certain things in common. And I use example of human and even chimpanzee that are more related than chimpanzee and the gorilla. You would think you will expect that gorilla and chimpanzee will have more things in common, but at the molecular level, we see that humans and chimpanzee have 98% sequence similarity. Right? Then we come back to the diversity level, you begin to see that even as much as we are related, even human beings, that okay, we are all humans, but we are not all the same. We also have our differences. So that is the diversity, similarity, and the diversity. So that is that for. So we have been able to cover two aspects of this topic, that is the concepts of biology and scientific inquiry. So we have covered two aspects. We have defined biology. We have looked at characteristics of life as one aspect. We have looked at unifying themes as another aspect. So before we move on to the last part, maybe we can take some questions. So maybe you can drop your questions in the chat room. I will pick randomly, and we move on. If anyone has any question, maybe let me visit YouTube as well and see maybe people have concerns that we can address. YouTube, good morning. Don't greet. Who are you greeting? Good morning, good morning. Good morning to all of you. So we don't have anything. I don't think attendance is going to be serious since admission is still going on. Okay, thank you. Demilade Giga, keep passing fake news. You are the one that will take attendance. That's why you are concluding. Please, attendance is compulsory. Attendance is compulsory and it's it's going to be taken seriously. You are the one passing information. Is anyone not understanding? Is anyone not understanding this, or is it only me? Anna Ayodeji, what exactly are you not understanding? You are on YouTube. Maybe you can private chat me on WhatsApp if there is anywhere you need us to elaborate on. When you said 98% similarity, do you mean chromosomes? No, not chromosome. We mean the sequences. What was it called now? The nucleotide sequence. When we do molecular, when we do DNA, you understand, remember nucleotide sequences. Like if you look at a protein, for instance, how does this protein differ in human, in goats, in dog, in gorilla, for instance? So you will see that maybe, for instance, at some points, maybe we have 10 points. Maybe you now have human and gorilla being similar at 8 or 9 points, whereas gorilla and goats maybe just 2 point similarity. That is what I meant by that. Then please, I don't understand what's the difference between DNA and RNA. We have not done DNA and RNA. When we go deeper into DNA and RNA, you will understand. How would we, let's see. Sorry, sir, is there any difference between DNA and RNA? Yes, there are differences between DNA and RNA. So when we go to DNA and RNA, we will delve more on that. So let's not use our time for biological, for another topic to take another thing. How do we chat you on WhatsApp? You have my number now. If you don't have my number, ask my representative. Ask your representative for my number. But please know what you are coming to my DM to say so that I will not block you. Okay. Let's go to Zoom and see whether we have questions there. Can you explain more on negative response? Okay. By negative response, we mean that, for instance, if it could be either way, negative response could be either way anyway. It could either be maybe something is too high, then there's a signal to bring it down, or something is too low, there's a signal to bring it up. That is, there is an opposite, negative feedback is an opposite direction reaction. Like, I will still use the same example on high temperature. Suddenly, if the temperature rises, right, then the body sends a signal that this temperature is too high, find a way to bring it down. Then if you now move from high temperature to low temperature, that is a negative signal, right? That it is bringing about the opposite reaction. So that's what I mean by a negative feedback. That is, there is high temperature, then the reverse is what the system really requires. So the system finds a way to nullify the effect of that high temperature by bringing it low or by bringing it down. So that is a negative feedback loop, and homeostasis works in that way. That is, if there are imbalances, right, maybe too high or too low, so the system sends a negative feedback that this glucose level is too low, start producing more glucose, right? Or this temperature is too high, find a way to bring it down. So homeostasis is a kind of, because we are coming to the middle. Look like, okay, imagine your what is it called? Your scale. If you have a scale, right? If it goes too high on one side and you want to balance it, what do? You tilt it up. The one that is low, you raise it up so that, or you add more to it so that it can be, for instance, this one is 10 kilogram, the other one is 8 kilogram. You see that the one that is 8 kilogram will go up on the scale. Then you need to add 2 kilogram to the 8 kilogram that you have to have a balanced equilibrium. So that is what we're referring to here in homeostasis. When something is too high, you find a way to balance it. When it is too low, you find a way to balance it. Either way, you are sending a negative feedback that this thing is too high. The negative feedback is that you must bring it down. If it's a positive feedback, you keep on increasing it, right? So that is a negative feedback, and I hope you get it now there. Okay, let me see, can we look at another question? Did humans undergo evolution? Did you undergo evolution? Difference between emergent properties and evolution. Okay, evolution simply means change over time. Like, there is change in organisms, or maybe speciation, new organisms arise. But emergent properties deals with organismal level, while evolution deals with is entirely about population, right, or group of organisms, right? Emergent properties that as you move from one level to another, it's just like when you are in primary 1, right? There are certain things that you cannot do until you get to primary 2. you now have certain properties, right? Like you don't expect to see features of primary 5 pupil, for instance, in primary 2 pupils. So that primary 5 pupil, we have more properties, certain features that helps us to cope, to survive in primary 5 than you now take primary 2 pupil to primary 5. So as you move higher level, you get certain properties, certain features that help you to cope, that help you to function very well in the level that you find yourself. So that is what we remember. As you move from higher one level to another, you have new properties, novel new properties that help you to cope, that help that organism to cope at that level. For instance, as you move from cell to tissue, you expect tissue to have more properties, to be able to do more certain things better than a cell. The same way as you move from tissue to organ system level. The organ system level, we have more properties than the tissue level. Whereas evolution, on the other hand, simply means how organisms have changed over time. And in fact, the definition of evolution is change over time. Change is different from properties, right? For instance, maybe you used to have some how do I call it, some insects, right, that are that are prey to certain birds, right? Maybe when they find themselves, maybe a dark a dark a light skinned insect, maybe the insect is light, right, light in complexion, that it is so obvious. Then over time, that insect or caterpillar or organism that was once conspicuous and easily seen by predator now develop certain traits or adaptation that make them to evade their predator, and over time those adaptation that is are now passed down to their offspring. So you call that natural selection. That is, nature is selecting traits that help an organism to adapt to its environment, and that adaptation is passed on to successive generation. So that is a adaptation and natural selection, right? So natural selection is like a coping strategy, right, for organisms. Okay, let me take one more question. Structure fits. No, it's not fits. So fits. F-I-T. Structure fits function. Structure fits function. Okay, let me type it. Structure fits function. Okay, I think we can move on now. Can emergent properties help with evolution? Emergent properties and evolution are two different things. Please stop bringing them together. Evolution and emergent properties are are separate entities, right? When you are talking about evolution, maybe you can talk about adaptation or natural selection. Emergent properties has nothing to do with evolution. Okay. If hemostasis is negative, what is the positive aspect? Maybe that will be an assignment for you. There are certain things that we are supposed to find out yourself to go and read. Does it mean homeostasis maintain or regulate give a negative feedback? What is it? Does it mean every homeostasis maintenance or regulation gives a negative feedback? Does it mean? Yes, every homeostasis is a negative feedback process. If it is homeostasis, it's negative feedback. Yes, the answer to Tijani Afusat Folashade is yes. Homeostasis is usually by a negative feedback. Okay. Okay, I think I'm done with the question. You can consult textbooks for further questions. We cannot take all questions like I said earlier. So let's continue. Okay, we have methods of inquiry in biology. So like we said earlier, we said that biology is the science that studies life, right? Then science, the word science comes from Greek word that means seeking knowledge, like you want to know. Please stop sending something in the chat room. Stop sending things in the chat room, please. The way it pops up, as it pops up, is causing distraction, please. Please. So we said biology is a science, right? Biology is science that studies life. So if it is science, I would say that science deals with knowledge. You want to know. Then how do biologists inquire? By inquiry, you mean what? You are asking question. You are asking question to know, right? So how do biology ask questions and how do they get answer to their questions? So that is what method of inquiry in biology is all about. That is how biologists find answers to certain questions, to natural phenomena, to observations, right? And that is what we are going to be looking at in the next 30 minutes. This class will go over 1 hour today, please. So kindly bear with us. We want to end this topic today so that maybe by Friday, God willing, we can move on to another thing. So the class might extend till maybe some minutes past 10 or at most 10:30, which should just be 1.5 hours. I don't think that is too much. Okay. Like I said earlier, the biologists use the scientific methods to study living world, and the scientific method is a problem-solving technique, right, that scientists use to conduct research to be able to get answers to life phenomenon, questions, and this is also applicable in our everyday life. And we are going to see how we apply scientific methods to our everyday lives. Scientists use two methods. Okay, in answering questions, in answering scientific questions, we use two logical thinking methods. We have the inductive reasoning and the deductive reasoning in biology. These two logical concepts are involved in scientific inquiry. Number one is inductive reasoning. Inductive reasoning simply means arriving at a general conclusion based on repeated observation. That is, you are moving from general to specific, right? You arrive at a general conclusion by observing certain things. For instance, if I say that the sun came out yesterday and today, definitely it will come out tomorrow, right? You are saying that, okay, using your knowledge of the sun, or maybe sun sets in the east, for instance, right? So the sun is always setting in the east, or because the sun came out yesterday and today, then you conclude that you reason by inductive reasoning that the sun will come out tomorrow. So that is inductive reasoning in biology. That is inductive reasoning. That is, you are moving the process of reasoning that move from specific observation to generalization, right? Or another example of inductive reasoning is that if I say, for instance, okay, how can I phrase this? Maybe you say cheap drugs cause, or maybe cheap drugs are ineffective. You said that cheap drugs are ineffective. Look, you now go to the market, you buy two type of drugs cheaply, then you find out that they are not effective. Then you now conclude that all cheap drugs are ineffective, whereas you have only sampled just two. So that is, you are making generalization based on specific observations. That is inductive reasoning. Whereas deductive reasoning uses a general principle or law to forecast specific results, right? So for deductive reasoning, you are moving, you are starting from a general statement to make a logical conclusion, right? General statement to make a logical conclusion. That is, you are moving from general to specific. You are moving from a general to specific, and this involves you carrying out certain research or hypothesis to be able to arrive at a particular conclusion. So that is inductive and deductive reasoning. Then, how do scientists study natural phenomena? There are two approaches to that. We can either use discovery science or hypothesis-based science. That is, don't forget, we are saying that the method of inquiry in biology is how biologists are able to answer questions, are able to profile, for instance, if you administer a drug on individuals, then you want to see the effects to be able to say, okay, this one was better than this. So that is how biologists are able to decipher, to make conclusion based on certain things. Then it can be maybe by discovery, maybe by using observation and data to arrive at a certain conclusion. Then we have hypothesis-based. This hypothesis-based talks about you are proposing and you are testing hypothesis to explain natural phenomena. For instance, if you enter a room, right? You enter a dark room. There are different observations, there are different hypotheses that may make that room to be dark, right? So number one might be maybe the bulb is bad, the switch is not working, hypothesis 2. Hypothesis 3, the lamp holder is not okay. Okay, hypothesis 4 might be maybe because you are going blind, right? So there are different hypotheses in hypothesis-based approach to scientific inquiry. That is, there are hypotheses that you can test to say, for instance, the dark room scenario. If you say that, okay, this room is dark because the bulb is bad, you change the bulb, or maybe because the light switch is not working. If you turn it on, or if you switch it on, if the bulb comes, that means your hypothesis, the switch hypothesis, is correct. But if after switching the switch, the bulb still doesn't come up, what do? You change the bulb, right? So all of these are different ways, and you do this, you carry out experiments to be able to do certain things. You changing the bulb is an experiment on its own, right? So we have discovery science, which use observation, whether directly or indirectly, to get data, whether qualitative data or quantitative data, to be able to arrive at a conclusion. For instance, when you hear data generally, what comes to mind is figures, right? Figure, data, no. Data are not necessarily figures. Data can be qualitative. For instance, if you observe, if you are studying an elephant, the data that you are getting might be maybe how frequent does this elephant visit a particular place? what does it do? You are not writing figures there, right? Although what you're also getting is data, right? So when you hear data, data does not mean figure. That's why data can be qualitative or quantitative. Okay, then the type of reasoning that we use in the discovery science is inductive reasoning. That is, you are moving from what's it called? From specific to general. Inductive reasoning moves from specific to general. Then we have a hypothesis-based science. Hypothesis-based use starts with observation. Like I said earlier, the observation is that the room is dark. Or let's move away from the dark room. Let's look at your torchlight, right? So the observation is that this torchlight is not coming up. Then you begin to wonder, what could go wrong with my torch? Then there are you may come up with two hypotheses there or question. When you observe, the next thing is that you ask questions. Okay, my torchlight is not coming up. Then you begin to ask questions. What is wrong with this thing? Is it that I bought fake? Is it that the bulb is not good? Is it different things, right? Is it the battery that is dead? So here you are asking questions, and your questions now form your hypothesis. For instance, if you think that it is the bulb that is dead, then your hypothesis becomes dead batteries or the bulb is dead, right? So to be able to, after the hypothesis, you predict that, okay, if I change the bulb, it should work. That is if, then. If I replace the battery, it should work. That is prediction. Don't forget we said hypothesis-based science starts with observation. When you observe that, why is this thing like this? You observe that something is this. Okay. You observe that your bulb, your torchlight is not working. You start asking questions. Then you form hypothesis based on your question. Then you predict. Then you carry out experiments to see what the problem could be, to be able to provide a solution. By experiment, your experiment actually now, scientifically, when you carry out an experiment, you're supposed to have a control experiment, right? The control experiment is, okay, we still delve into control experiments. Let's not go there now. So you make prediction that, okay, maybe, like I said earlier, replacing the battery could solve the problem, or replacing the bulb could make your torchlight to come up. Then what you do from there is that you test your hypothesis, right? By testing the hypothesis, you are carrying out your experiment. And that is, you are replacing the battery or you are replacing the bulb. Then you collect data scientifically. Now, when you are doing scientific research, you collect your data, you analyze your data. For instance, if you want to, maybe you are hypothesizing that maybe drug A works better or works more effectively than drug B, right? So what do? You administer both drugs, drug A to some certain group of individuals, drug B to another group of individuals. Then you have your control group that you don't administer your drugs on. Then you collect your data and be able to analyze, okay, from among those that you administer drug A, maybe 10 groups, how do they react to the drug? Then from the 10 groups in drug that you administer drug B on, how do they react to drug B? So then you collect your data, you analyze, and when you are doing such research, you're supposed to have the groups, like I mentioned there, equal number of individuals per group. Not that you have one group, 5 individuals, another group, 10 or 15 individuals, and you are saying that. So that is not how to do research. If you are using 3 individuals, use 3 constant number for the control group, for the treatment groups. Then From there, you can now make your deductive reasoning that, okay, from my experiment, this is what I can deduce. Then you conclude. Okay, so from there, you can now conclude that based on your research, based on the research I've carried out, this is the conclusion that I can arrive at. Before we proceed, let's see, do we have any questions? Any question? If you have any question, maybe you can raise it now. Let's take more questions before we proceed on a scientific inquiry. We said the scientific inquiry tests how scientists or biologists are able to provide solution to answer certain questions. Right? We said the word science itself is asking questions. You want to know, you are seeking for knowledge. Science means seeking for knowledge, to know. That is what science is all about. Then we said by science use two logical thinkings to answer questions. So we have the deductive reasoning and the inductive reasoning. Okay, people are asking, what is inductive reasoning? What is deductive reasoning? We said that deductive reasoning, inductive reasoning means you are arriving at general conclusion based on repeated or related observation. That is what inductive reasoning, excuse me, is all about. That is, you are arriving at a conclusion based on repeated observation, right? So, and I said that by inductive reasoning, you are saying that, okay, maybe because the sun came up yesterday, then, and today, so you predict that it will come up tomorrow. So, inductive reasoning is not how do I put it now? It's not definite, right? The conclusion is probably is other probability is probably true, right? Please don't drop what's it called? Anything in the chat room. Right. So inductive reasoning simply means you are concluding, right, based on general repeated observations, right? You observe that the sun came out yesterday and today. Then your inference is that the sun will come out tomorrow. So it's an inference. You are inferring, right? That in inductive reasoning, you are inferring that because this occurred today, then today and yesterday and today, then tomorrow it will be so, based on your repeated observation. Then you are arriving at a probabilistic conclusion, if I can use that word. Like, okay, the sun will probably come out tomorrow as well. But in deductive reasoning, it's more logical, right? We are using logical process, using accepted facts to draw conclusion, right? Like we say here, using a general principle or law to forecast specific results. For instance, and it is expressed using like what we know as is based on premises, like if A, then B, right? And the premises have to be true. One premise has to be true for the other one to be true in deductive reasoning. For instance, if you say one premise is that maybe, okay, for instance, if you say how can I explain this? If something, sorry, Okay, we said deductive reasoning is a form of logical thinking that uses a general principle or law to forecast specific results. So by deductive reasoning, you are is based on premises like if this is this, then you can conclude this way. I don't know if you are getting it. A reasoning that, okay, if we said that such reasoning it moves from specific observation to broader generalization. Like you are moving from specific that if this is this, then you expect the other way to, for instance, if okay, let's use our dark room and bulb as an example, for instance. Like if you enter a dark room, then you want to believe that if you change the bulb, then the light should come up. That is one of deductive. You are deducing by experiment, by law that says that, okay, if I run out of fuel in my car, for instance, then if I buy fuel into it, then my car should work. But one premise has to be true for the other one to be true. For instance, your car that suddenly starts working, maybe because you run out of fuel. Then if you bring that fuel is true in that regard. There is fuel, then you can see that if you power it up, all of that things being equal, your car can move. So the deductive reasoning is moving from general principle to specific result. The general principle is that if you supply a car with fuel, the car should work. So that fuel is your specific result that the fuel is the specificity in that explanation. Okay, let's see if we have another question. All Africans are black, and you say Kenya is African, then is black, right? Deductively. What is this? Is that okay? What is the constant? First, please, I'm in the chat room. I'm trying to see your question or comment. Explain hypothesis. I mean the hypothesis before prediction. Can you buttress more deductive reasoning? How does deductive reasoning work? Explain deductive reasoning on these steps, sir. Explain the deductive reasoning. Deductive reasoning. Inductive and deductive. Okay, I think we're still having problem grasping okay, somebody said it, like if you say all Africans are black and you say Kenny is African, then Kenny is black since Kenny is African, right? Deductively. This is inductive reasoning. It's not deductive. Like, for instance, if I say all students smoke, and because I see one of you smoke, and I conclude that everybody is a smoker, right? This example here is a inductive reasoning by observation. By observation. This is inductive, not deductive. That is Ogumbu Mikende. This is inductive reasoning, not deductive. Okay. Okay, so discovery science use observation and data. And okay, hypothesis. Okay, somebody said, well, I should elaborate more on this. Okay, by hypothesis-based science, hypothesis-based science starts with observation. You need to first observe. For instance, you observe that this room is dark. You enter a room and you observe that the room is dark. Then you begin to answer questions. Then you begin to ask questions. Why is this room dark? Right? Please, so can we stop this explanation? Please. Please. your comment in the chat room, the way it pops up on the screen distracts. Oh, please stop typing in the chat room. If you drop anything again now, I will remove you. Okay, we said accurate observation. When you observe, right, then you begin to ask questions. Then from your question, you formulate hypotheses. Right? Hypothesis is an educated guess, right? You are guessing that, okay, this thing is not working. Then you guess that if I do this, it should work. Then you predict that, okay, if I change the bulb, the light should come up. Then you carry out your experiment. By experiment, you are experimenting. You are carrying out experiment based on your hypothesis. Like, for instance, you want to look at effect of fertilizer on plant growth. What do? You carry out an experiment. So that is experimenting. Then from experiment, you collect data and you analyze to have your results. From your results, you can deduce. Then from there, you can conclude. That is the hypothesis-based science, and that is illustrated more here. Observation, then down to question. Then there can be several hypotheses. It doesn't have to be one hypothesis, like I said earlier, using the example of the dark room. It can be the bulb, it can be the switch, it can be the lamp holder, it can even be electrical connection. Maybe the wire somewhere is faulty, right? So you can have more than one hypothesis. There may be several hypotheses. Then you subject them to experiment. Then you begin to reject. Maybe out of the 5 hypotheses that you start with, you have rejected hypothesis 1 and hypothesis 5. You are left with 3 hypotheses, maybe hypothesis 2, 3, and 5. Then you carry out further experiment on your hypothesis 2, 3, and 5. Then you reject 2. Out of the 3, you are left with 1. So hypothesis can either be accepted or rejected, right? So when you reject one, you accept the other. So when you reject, you now have one hypothesis, then you carry out your prediction based on your hypothesis. Then you carry out several experiments, you repeat experiment. Then you modify hypothesis. From there, you can now confirm your prediction that, okay, of all the 5 suggestions in quote, or 5 guesses that I make or that I propose to this problem, I was able to carry out a research on all and find out that this is what is actually wrong. It's just like when your car breaks down on the road, right? Then maybe you call a mechanic, then they begin to say, okay, when did you change the fuel? What did you do? Do you know? So all of this then begin to make just like when if you visit a doctor, for instance, then they're asking you question, what is wrong with you? So by answering the question, the doctor begin to eliminate certain guesses. Could it be malaria? Okay, it's not malaria. Okay, could it be this? What could be this? So that is what, then they now go finally to go and carry out a test, a form of experiment to be able to conclude on what actually the problem is, right? So that is deductive science or hypothesis-based science. You are making a hypothesis, you are testing your hypothesis to be able to conclude that, okay, you have a fact, right, to say this is what is actually wrong. Now, not just saying things by fallacy, right? But you have facts, you have data, you have carried out your experiment, you have data to back up your claim that this is what is actually wrong. Okay, so let's look at those steps. Maybe we can now understand it better by observation. So an observation can be defined as seeing something with your eyes or a new idea that comes by reasoning. You may want to just reason, okay, what if I do something like this? What could, just like some people have ideas to produce electricity. These are ideas. They don't see, they know that they see electricity like this, right? It's an idea that come up. Then they go to the lab and experiment. Okay, if we do this, if we put this together, what are we going to get, right? Somebody said that there are the light experiment. There are thousands of failed experiments, right? Before they realize one that works, right? So all those ones are hypotheses. These hypotheses were tested and they were rejected because they see that they were not, it doesn't support their guess or their claim. So you begin to test several hypotheses when you have those ideas that come. So that is observation, what you see. For instance, you see that your bulb is not, your torchlight is not working, your fridge is not working, then your car broke, what could be the problem? You observe certain things first. Then you make educated guesses in form of hypothesis. Okay, maybe it is this or that. Then from observation, you formulate hypotheses, like we said earlier. So hypothesis for a natural phenomenon, or all hypotheses are testable. That is, you must be able to put them to test. For instance, if you say salt dissolves, that is your hypothesis. You must put that salt in the water to be able to see whether it dissolves or not. So you must be able to carry out a test on your hypothesis. Don't go and make an hypothesis that you cannot test, right? So hypotheses are testable explanations. They are formed based on observations. Like I said earlier, you must observe. Hypotheses don't come before observation. Hypothesis. You observe that something is wrong with your system, and then you be, okay, what is the, decide that, okay, if it is this, then this should work. Then hypotheses are either accepted or rejected. You don't prove an hypothesis, right? Because you only accept it. When you accept your hypothesis, it means that what you propose, what you think when you carry out your research, and for instance, when you change your bulb, if the torchlight comes on, then it means that you accept the hypothesis that it is the dulled bulb, right? Then if the light does not come on when you replace the bulb, then you reject that hypothesis. But if the light comes on, you accept hypothesis. Hypotheses are never proven. They are only accepted or rejected. Then, like I said earlier, all hypotheses are testable and falsifiable. I've explained testable, but by falsifiable means that hypotheses can be proven, can be wrong, right? They can be false, right? That is, for instance, if you say the bulb is bad, if I change my bulb, my torchlight should come up. Then when you change the bulb, the light doesn't come on. That means that hypothesis is falsifiable. They are not always true. Hypotheses are not always true. They are falsifiable and they are testable. The hypotheses guide the design of your experiment, right? The hypotheses that you have formulated determine how you set up your experiment, how you design your experimental setup, right? So that is that on hypothesis. Then we have the scientific method, testing of hypothesis. Number one, you explore a phenomenon and make observations. You construct a question to investigate based on your observation, right? Like we have illustrated before. You construct a hypothesis. You state your prediction based on hypothesis. You carry out your experiment. You collect your data and analyze them. Then make a conclusion. And you are now profiling knowledge, which is what science is all about. Science is about knowledge, right? When you do a research, what you are doing is that you are contributing to knowledge from your research. For instance, you want to look at effect of heavy metals on plant growth. So by your research, your finding is a contribution to knowledge. Okay, maybe certain heavy metals affect plant growth or maybe they even elucidate parts in the plants. So all of these are how scientists, biologists, scientific knowledge by into the future, and they ask questions and they are able to provide solution and they arrive at a conclusion. Then you can give knowledge to your prior knowledge and the knowledge of others, that is existing theories. For instance, if you carry out a research on which example can I use? Maybe diversity of a particular plant in Nigeria or in Lagos, southwest Nigeria. Diversity of maybe Terminalia catapa or a particular plant in southwest Nigeria. So that is your own finding. You may now want to relate your finding to what somebody did already in Nigeria. How diverse the diversity of this particular plant in Jos, in Nigeria, in Cameroon, in Germany, relating your findings to what is already known in the field. Then you may still want to identify gaps in literature. That is follow-up question for investigation. For instance, if the diversity of that particular plant in Nigeria is going this way, in Lagos is going one way, then you begin to ask questions again. So that is where we say there is no end to seeking knowledge. The day you stop learning, you are dead. So if you find something today, you may want to ask again, why is my result different from not do correctly? Then from there, you are laying foundation for future research, future knowledge. That is, you are going back to the stage of asking question again, right? So that is how science that's how the scientific research is conducted to be able to arrive at conclusion and provide information to natural phenomenon or solution to. Okay, before we go further, do we have any question? We are coming gradually to the end of the class. So we can take, this is, what time is this? 10:21. Okay. Sorry, let me go to YouTube. Let me go to YouTube again and see what is happening there. So that won't be as if somebody said people are not serious. Somebody was referring to YouTube people as lost people. They are not lost too. They know just that they know that you join early doesn't mean that. Can you explain more on emergent properties? Wow. Haven't I come here a long time? We left emergent properties in 2021, 2025. Okay. Maybe as a recap at the end of the class, we can go back to emergent properties. Okay, I think hypothesis is a guess on why prediction suggests a solution. Okay, that is another understanding of by somebody. Okay, I don't think we have any question here on YouTube. Let's go to Zoom to pick one or two questions before we move on. Which is more reliable, deductive for it? We have left deductive reasoning. It's a must. What is it? Is it a must? Okay, let's say, is it a must? We see the observation. Can't we feel it? Maybe through touch or other sense. Said you can either see it with your naked eye or you imagine idea. It could be an idea. It's not be something that you see. It's there in the slide that it could be observation is here now. it could be as saying something or a new idea. You can't see an idea now, my dear, that has that question or that put it there. That is a Tiamiyu Abdusalam. So it could be an idea. You don't need to see it though. You don't need to, it shouldn't be what you can see. Is hypothesis idea based on imagination? Imagination and hypothesis. No, hypothesis is different. Your imagination is different from your hypothesis. Your imagining is different from there is an observation. If you are imagining it, is you are not observing it. Imagination is even different from idea, right? So it's imagination and hypothesis they don't go together, please. Can hypothesis become a theory? We are going there. You were taught in secondary school now. Why are you asking me, can hypothesis become a theory? You want to know. They told you in secondary school, yes or yes. Okay. Can we relate hypothesis as tests? Can we relate hypothesis as tests? What is that question? What is that person asking? Hypothesis, tests, experiment. Hypothesis will lead to testing something now. Like for instance, if you hypothesize that injection works better, test both injection and drug on the same individual to be able to see that. Is hypothesis the same as prediction? Hypothesis leads to prediction. Hypothesis is not the same as prediction. Hypothesis is you are making a guess, right? Then from there you predict that if you do the happen. They are related, but they are not the same thing. Okay, let's go back to our class. I don't really how you group the whole thing. You don't have to go. Who is this? Somebody is saying something. I don't need to get. I don't understand what you are saying. Are you saying I don't have to explain? I don't. Sorry, please. Okay, let's continue with stuff that hypothesis. We have looked at this. We have also looked at this. Okay, so we said from hypothesis, you carry out your experiment. So experiments are exercises designed to test the hypothesis or prediction made by the hypothesis, right? So you have made a guess, right? You have hypothesized, you have predicted that if you change your bulb, your light should come on. Then you do the experiment. The experiment is now, for instance, if you say that people that consume fat and oil more are more intelligent than people that do not. Like you are saying that people that consume certain food or nutrients are more intelligent than others. So what do? That is just a guess. That's an idea, a reasoning that is coming to you that people that do this, so you need to test it scientifically. Like I said, don't just make a blatant statement in science. That's why I say, what is the fact? Where is the data to support your argument? So you need to do experiment to get data. In science, we rely on data. If federal government tells that unemployment is reducing, where is the data? How many graduates do we have? How many jobs are produced every year? How many? And that is how you arrive at conclusion. So science speaks to data. Okay, and how do you get your data? Is by carrying out an experiment. You don't cook figures, you don't cook data. If you are a scientist and you are cooking data to support your guess, then you are a bad scientist. And you can even go to jail for that. Okay, so experiments are conducted to test what we have proposed, what we have guessed as a hypothesis. Then It is also known as the data collection step, whereby you are collecting data to support your claim, right? Then, where possible, experiments should contain a control group. That is, when you are carrying out an experiment, you're supposed to have a control group. Who are these people still sending something on deductive reasoning here? So, what is a control group? A control experiment, sorry, I think the way I arranged this slide, the control experiment, we said that every experiment should have a control group. A control group is where the is missing. Come back here. Okay, so that where possible, experiment should contain a control group. So control groups are individuals that a group, right? That you alter a factor that you are testing. For instance, if you say that people that consume fats and oil are more intelligent, so you have control, you have groups. One group gets fat and oil. They are the experimental group, because that is what your prediction is based on. That if I feed you with fat and oil, you are going to be more intelligent. Then there's some other group that you don't feed with fat and oil. That group that don't have fat and oil is the control group. We can have positive control group or negative control group, right? Negative control group is where one of the variable is absent. That is, your test solution or your experimental setup is absent in negative control. For instance, if I want to see whether a particular drug will induce cancer, so I get my lab rats or mice. One group, I give them the drug that I think will induce cancer in them. Then the control group, I don't give them the drug that I proposed or that I guess that will induce cancer. So the group that does not receive that dose is the negative control group. That is, you don't expect any response as a result of the treatment in them. But positive control group, positive control is when you treat them with what we, for instance, maybe you are looking at effect of a particular vegetable on cancer treatment, right? You are looking at effect of plants on cancer treatment. So there is a group, that positive control group, that you, after inducing your cancer, right, you will not give them the remedy that you want to test out, right? In positive control, you are giving a treatment that will elucidate the positive in such a way that the results in the tested group can be adduced to your test solution or test material. Another example is that, for instance, if I want to see, if I want to see whether paracetamol, right, can induce cancer. I just want to see. It's not possible, right? I'm just saying that I want to see whether paracetamol can cause cancer. I will have my treatment group that I will induce them or inject them with paracetamol. Then I will now go and look for a known material or test substance that I know that this particular drug has been tested and proven to induce cancer. So that particular drug that induces cancer, I will now administer it on my positive control group. So I will now say that, okay, this is as a result of the positive control group will elucidate the what is it called now? The expected outcome for sure. It is shown in them. So that is a positive control in scientific case. Then when you set up your experiment, there are certain variables to look out for. It is those variables that you analyze in form of your data to be able to arrive at a conclusion. We have the independent variable. The independent variable is the factor that is being manipulated by the researcher. That is independent variable. You are testing, you are what is it called? Those things that you can alter by the researcher. They are what the researcher can alter. For instance, you want to look at effect of fertilizer on plant growth or effect of fertilizer treatment on crop production, right? Whether fertilizer increases crop yield. So what can you do? Your independent variable in that experiment is your fertilizer, because you can alter it, you can manipulate. That is, you can have a group that you put fertilizer, you have another group that you don't put fertilizer. So the variables that the researcher can manipulate, can toy with, is your independent variable. Then we have the dependent variable. The dependent variable is the outcome that is being measured by the researcher, or the outcome that you are recording by the researcher, and you predict that it is whatever you are getting in the independent outcome in your independent variable. In your dependent variable is a factor of the independent variable. For instance, going back to our example on effect of fertilizer on crop yield, after applying your fertilizer to a pot or to your plant, then you are now seeing, okay, how is the yield? Okay, how many bushel of corn am I able to get when I apply fertilizer? So you can even vary the quantity of the fertilizer that you apply, right, on the same pot of on the same plot of land. So the independent variable is usually what the researcher can manipulate, and in most cases, it's usually one. You are not expected to have several independent variable at a time. You're supposed to have one dependent variable so that you can really say that what you are testing is as a result of what you are getting. What you are getting is as a result of what you are, because if you have several independent variables, you will not be able to know what is really giving you the output or the outcome. So it's usually one independent variable is tested per time or per experiment. Then the dependent variable is the outcome as a result of your manipulated independent variable. I hope that is clear. You can say the independent variable is the one that you can manipulate. The dependent variable is the outcome or the result that you are measuring as a result of that is affected by independent. Then you have the standard variables. Standard variables are all other factors that remain constant. For instance, in our experiment on effect of fertilizer on plant yield, on crop yield, other standard variables might be water. You supply them with equal amount of water. That is your water. There's a standard variable, not that the one that you treat with fertilizer, because you have supplied fertilizer, you don't put water. No. The standard variables has to be there is equal amount of water for them. You put both of them under the same condition, not that you keep one in the wardrobe and keep the other in the open air, right? So these standard variables are all other factors that are constant, that are the same in all of them. It is only your independent variable that is different. For instance, effect of fertilizer on crop yield. You have two pots or two plots of land. One, you apply fertilizer. Fertilizer is your independent variable. The other one, you don't apply fertilizer. The other one, you don't apply fertilizer, is your control group. Like we said earlier, that all the experiments you have usually have a control group, right? Then your standard variables are all other atmospheric conditions that should be constant in both. You supply equal volume of water, you expose them to sunlight and what have you. Then let's look at these variables in experiment, independent variable versus dependent variable. For example, we want to look at effect of room temperature on test scores. Here, your independent variable is what? Your temperature. Don't forget, we said the independent variable is what the effect is seen in dependent variable, right? So the independent variable here, like we said, effect of fertilizer on crop yield. Here, effect of temperature on test scores. Maybe people that write exam under different conditions. Maybe I put some of you inside AC to write your exam. I keep some of you in the sun to write exam. I keep some of you under normal room temperature to write your exam. And I want to see how that result compare among the several groups. So my independent variable in that wise become the independent variable is the temperature, and the dependent variable are the test scores that you see. So the independent variable is the cause, while the dependent variable is the effect. You can manipulate the independent variable, but you measure the dependent variable. Then when you want to plot your graph, you are plotting your independent variable on the x-axis, and you plot your dependent variable on the y-axis. Who is that is saying Omo? If I see you, I will block, and I block you forever. What's enough that you are saying in my group? Make una use ChatGPT. So Puruchuku David or Swagu. Make them use ChatGPT, Abi. No problem. Okay. So like I said earlier about control experiments, I said experiments usually have positive control and negative control, depending on what you are looking at. The negative control is where all the steps are present, but only one factor, that is the factor that you are testing, is missing, while the positive control tests that give a known positive result are also appropriate for some results. Experimental controls provide a reference for comparison, so you can be able to say that what you are seeing, your results are reliable. We've dealt with negative and positive control. Then scientific methods, then down to conclusion. From there, from your results of observation and experiments are analyzed and conclusion reached, the conclusion is either the rejection or hypothesis in step 2. In step 2, if the hypothesis is rejected, a new hypothesis is formulated. Then you go back to step 2. If the original hypothesis is accepted, the original hypothesis gains strength. If it is rejected, you form a new hypothesis. Like I said earlier, hypotheses are either rejected or accepted. So when you reject one hypothesis, you accept the other. Then further positive testing and evaluation may lead to hypothesis progressing to scientific theory. Somebody asked that, can hypotheses become theory? Yes, hypotheses can become theory after they are tested and validated, right? Then they become a scientific theory. Hypotheses are really accepted or rejected with absolute certainty, because that is what we meant earlier, that by hypotheses are not proven. They are either accepted or rejected at the point that you are making that findings. It might be that maybe tomorrow, the hypothesis that you postulate, another person can come tomorrow and say it is not so. Like John Baptist Lamarck, in his theory of natural and acquired of inheritance traits, Lamarck's theory of evolution, he hypothesized that organisms can transmit acquired traits. Like for instance, if you develop biceps because you carry weight and do gymnastics and what have you, then if you acquire six packs rather, then your children all of them will have 6 packs. But when it was put to test, it failed the test of time. Then hypotheses are, it becomes falsifiable, like I said earlier. So you don't say with absolute certainty that this hypothesis is accepted with utmost certainty or rejected with utmost certainty, because tomorrow an event may catch up, and what you say works today might not work tomorrow. In fact, some other variables, like we now have a mosquito insect, what's it called? Insecticide repellent mosquitoes, right? That resistant mosquito rather. So when they first did those insecticides, your bacon and what have you, when you feed palm, they die. Then you can decide that if I feed this mosquito, it will die. Then over time, they develop resistant genes that made them to be resistant to fleet, to bacon or what have you. Then when you fleet them, they don't die again. Then you go back and do another research. So that is why we are saying that you don't accept or reject a hypothesis with absolute certainty. And hypotheses that has withstood the test of time and evaluation by numerous experiments generally become a theory. Yes. Then theory explains the vast amount of data and observation with a relatively simple concepts. Theories are never set in concrete and are likely to change over time. The only thing that is constant is a change. So what you say is constant today. What you say is this today, tomorrow may not be so. And with that, we have come to the end of the first lecture or first topic in BIO 101. Will a theory lead to law if accepted hypothesis leads to... What is this person saying? Already rejected hypothesis be used if the accepted one is proven to be wrong? No. If one hypothesis is rejected forever. For instance, if you change your bulb, it doesn't work. It's not probable. That might not be the problem. That is why I said that there might be several hypotheses. I think we use one slide that has 5 hypotheses and scale down to 1, right? Once hypothesis is rejected, you go and test other hypotheses. So what is positive control group? A positive control group are those sets that you know that a test that will give known positive results. The groups that you know that, okay, this is what I expect you. For instance, you want to see whether paracetamol can induce cancer. Then you treat some group with paracetamol, your experimental group with paracetamol. Then the positive control group, you now test them with a known carcinogenic, like chemical, a chemical that you know that we cause cancer, we induce cancer. So a positive control is a group that you expect to see the groups you expect to show you the expected results, right? Because, for instance, I don't know, I want to kind of experiment that. Okay, maybe how do I put it? I will still use my cancer analogy. Like, you want to know whether a particular drug can do certain things in the body. You are not sure. Then you need to have a positive control group that's a known substance that has been proven to show what you are looking for. That group that shows expected results is the positive control group that give you positive results that you are looking for, is the positive control group. Why the negative control group? Test substance, your independent variable is missing. That is the negative control. The independent variable is missing in them. Okay, we have come to the end of today's class, and we are done with one topic already, and we still have some more to cover. Thank you for your time, and please, some people are asking, how is attendance taking? How is attendance serious? Please, attendance is serious. And we are taking attendance. All right. Do have a wonderful day. If you have further question that you think you cannot answer on your own, but I still believe there are no questions that you cannot answer on your own, maybe you have my number. You should have my number. You can ask those people that send the link to your group for my number. They will give you. But if you have my number, don't come to my DM and be saying what will make me block you. Comport yourself in a good manner, right? Okay, thank you, and enjoy the rest of the day. Our class, we have almost 2 hours. Bye-bye.